Unit 3, page 2, enzyme structure. Do now identify the organic compound for each example. You should have come up with carbohydrate. An example would be glucose, nucleic acid, an example DNA, carbohydrate, an example is starch, and protein, example enzyme, lipid, example oil. A catalyst is any substance that alters the speed of a chemical reaction. This includes things like heat, light, and in our body, enzymes. Chemical reactions are needed for all living things. Digestion means to break down. Synthesis means to create or put together. Chemical reactions create new substances. A reactant yields, that's what the arrow stands for, a product. Reactants are the raw materials or ingredients of a chemical reaction. Products are formed from the reactants. The arrow means yield. In living things, this is done by enzymes. What are enzymes? Most enzymes are proteins. They act as catalysts to alter speed of a reaction. They are not permanently changed or used up in the process, and therefore they are reusable. Enzymes are a specific shape. So we can see here a protein molecule that is in fact an enzyme. Enzymes are specific for what they will catalyze because of their shape. Their names often end in ase, like amylase, lactase. The enzyme is the green molecule that we can see here. It contains a shape known as an active site. The active site is a specific shape that fits the substrate. The enzyme-substrate complex. The substance or reactant an enzyme acts on is the substrate. We have our enzyme here, and it acts on the substrate. So the substrate will join the enzyme in the active site. The active site is a restricted region of an enzyme molecule which binds the substrate. So we can see here again our enzyme. Its active site would be where? Exactly. And it would connect with a substrate. Enzymes are a specific shape and reusable catalyst that alters the speed of reactions and are not changed in the process. They end in the term typically ASE. So in summary, what's another name for a biological catalyst? An enzyme. Next question. What process controlled by enzymes breaks things down? Digestion. What process controlled by enzymes puts things together? Synthesis. Explain why an enzyme works on a single substrate. Shape determines function. The active site can only fit one substrate.